Now, up to now we have just discussed how the macrophage take the antigen, process the antigen with class 2 molecule, then present the antigen along with the class 2 molecules to appropriate T cell. Appropriate T cell is a T cell which has such TCR which could recognize this specific antigen, and this T cell has CD4 molecule which will confirm that class 2 molecule is present with the antigen or not. Once TCR and CD4 are properly occupied, they stimulate CD3 molecule which through multiple molecules give signals to the nucleus of the T cell. This is a selected T cell. Meanwhile, macrophage also releases interleukin 1, right, and which is stimulating the interleukin 1 receptors in the T cells, which also stimulate the nuclear machinery of the lymphocyte. Moreover, B7 molecule which are expressed on the surface of macrophages interact with the CD28 and also give signals to the nuclear machinery. This is called the first signal and this is called the second signal, or we call this is primary stimulation of T cell and this one is co-stimulation of the T cell. Once a T cell has been properly selected and properly co-stimulated, then multiple genes are activated within the T cells. First of all, the group of gene which is activated, they are concerned with interleukin 2. One gene start making such messenger RNA which will be translated into interleukin 2, and these interleukin 2 molecules are thrown out of the T lymphocyte. But at the same time, another gene is expressed and that gives order to make such proteins which are actually interleukin 2 receptors. So interleukin 2 receptors are also, these receptors are also expressed on the surface of the selected T cell. So interleukin 2 which is secreted auto-act in an autocrine fashion on the interleukin 2 receptors which give signal to the nuclear machinery back, and then nuclear machinery activate mitotic mechanisms, and this selected lymphocyte repeatedly undergo mitotic signals and mitotic activity, and it makes clonal expansion. Is that right? Let's stop the story here. We'll talk about now another, for a while we'll stop the story here. We'll link the story, first story with the second story later. Now we start the second story. Have you heard of the B lymphocytes? Right? Let's see meanwhile how the B lymphocytes are activated. Right? Do you remember your irresponsible friend neutrophil? What it was doing? It was taking the antigens and throwing into circulation, though not into tissue fluid, and those anti-free antigens through the tissue fluid passing through the lymphatics were going to the lymph node. Even free antigens are not wasted. B cells love to react with free antigens. Now let's see. What are B lymphocytes? B lymphocytes are those lymphocytes which mature into bursa fabriceus in the bird, or equivalent of bursa fabriceus in human. In human beings, equivalent organ for bursa fabriceus is our bone marrow. So B lymphocytes mature into bone marrow, then they come into circulation, and some of them settle into lymph nodes. Now let's suppose these are 5 B lymphocytes. 1, 2, 3, 4 and 5. These are 5 different B lymphocytes. Now listen carefully. Every B cell is genetically pre-programmed to produce a unique type of antibody against only a unique antigen. Again, listen carefully. It's worth revising. Every B cell is genetically pre-programmed to produce a unique type of antibody which will only act with a specific type of antigen. It means that different B cells, B lymphocytes, are pre-programmed for the producing antibodies against same antigens or different antigens. Again, listen, you are confused. Listen. For example, there are, okay, come here. This is our lymphocyte. Just you also come. I need 4, 5 B lymphocyte. This is one B lymphocyte. This is second B lymphocyte. There's a third lymphocyte, B lymphocyte, and here is, yes, you go there. This is 5. These are 5 B lymphocytes. Now, what every lymphocyte is doing? Every lymphocyte is expressing the sample of the antibody which it can make. Don't look at my head, it's bad. Look at my hand. Right? Now, actually every B cell is pre-programmed. The nuclear machinery of this cell is already pre-programmed to make a unique type of antibody. Nuclear machinery of this B cell is pre-programmed to make a different type of antibody. And we have millions and millions of B cells which have a capacity to produce slightly different antibodies against different antigens. Is that right? Now listen carefully. For example, this B cell is pre-programmed to make anti-star antibodies. This B cell is pre-programmed to make antibodies which are anti-moon antibodies. This B cell is pre-programmed genetically to make anti-sun antibodies. And this B cell is pre-programmed to make anti-dog antibodies. Is that right? If dog never comes, this B cell will never work. But if dog again and again comes, it will make again and again anti-dog antibodies. You understand what I really mean by this? Different B cells are pre-programmed to make antibodies with a unique specificity against a unique antigen. Is that clear? Now, what every B cell is doing? Every B cell synthesizes some of the sample antibodies and put these antibodies on the surface. Bring your hand. Okay, so this is, okay, he's making like this, here stark and fat. You make a different type of, no, it's, you can make it simply open. Yeah, this is anti-moon. You make it like, yeah, his antibodies are for anti-sun, and you make it like this. These are anti-dog. So what happens, these are 4 lymphocytes, on their surface they are expressing different type of antibodies. This is a molecular message to different type of antigen. Now, star antigen, neutrophil has eaten the bacteria, released the star antigens. Stars are coming like this. Do you think star will fit into dog system? Sorry, dog, anti-dog antibody system? No. Will they fit here? No. Will they fit here? No. Star antigens will truly fit here. Right? Now, as soon as it becomes fit over here, this B cell becomes very happy that my antigen has come. Is that right? So this B cell will be selected. Again, there are soluble antigens. B cells are very humble cells. They don't say that if gift comes, someone should come with the gift and wrap it properly with class 2 molecule, nothing. They say even someone throws it, we may accept it. Is that right? These are humble cells. So what really happens, that neutrophils were eating up the bacteria and throwing the bacterial antigens into fluid. Through the fluid, those bacterial antigens also reach to the same lymph node where the macrophage had personally go to activate T cells. The free antigen sooner or later will interact with the sample antibody on the B cell. Right? Which will be that antibody? Anti-star antibody. These antibodies, please express your antibodies on the surface. These surface antibodies are called B cell receptors for the antigen. These surface sample antibodies are called B cell. What are these? B cell receptor for the antigen. Is that right? So what we say, every B cell is expressing BCRs as T cell were expressing TCRs. So B cells have BCR. The difference is that TCR bind only peptide. BCR may bind with peptide or even with the carbohydrates. You know, they're humble. They may bind even with lipid, even with DNA. So BCRs can bind with variety of antigens. But TCRs as a group, they only bind with peptide antigen. Then BCR bind even with the free antigens. But TCR bind only with those antigens which are processed with MHC molecules. Is that right? And they only react T cells when macrophage or other antigen presenting cell in person come. So do you think which cell is more humble? B cell. Which is arrogant? T cells. T cells are very arrogant. They want the antigen, but they want that antigen should be brought in person by the antigen presenting cells, and antigen should be properly presented with class 1 or class 2 molecule. T cells are arrogant lymphoid cells. B cells are very baby type. You know, they are very, very humble. Right? They are not very arrogant. B lymphocyte, they have the BCRs. They may accept the free antigens. No one can come into person to give them. They may even accept not only peptide, they may accept carbohydrates. Some Some BCRs may accept carbohydrate, some other BCR on some other cells accept the peptide antigens, some other BCRs on some other B cells may accept the DNA or nucleic acid antigens, but T cells, TCRs accept only and only peptide antigen. It means if antigen is not peptide, T cell will not play the game. Is that right? Clear to everyone? Now think of about our star. Our star antigen came to this, no interaction, no interaction, no interaction. But this is happy, star antigen fit over here. Now you people go to your seats. We have selected this B cell. This is the selected B cell. Is that right? Now I think this B cell should also be asked to sit, and we'll draw the diagram. Thank you very much to everyone. Now what are these? These are BCRs. What are these? BCRs. Right? Different BCRs are for different antigens. Actually, these are also called surface immunoglobulins. Later on, I will tell you the surface immunoglobulins which are present on the B cell, which are functioning as BCR. Surface immunoglobulins are present on the B cells, which are acting as functionally as BCR, B cell receptors for the antigen. These are surface immunoglobulins or surface antibodies. Classically, they belong to IgM class or IgD class. I will explain this later in lectures. Right? There are 5 main classes of antibodies. Right? There are immunoglobulin M, immunoglobulin D, then there's immunoglobulin E, G, and A. On the surface of the B cell, either there are immunoglobulin M or there is immunoglobulin D. Right? So we can say that surfaces of the B lymphocytes are decorated by the surface immunoglobulins which are belonging to the class of IgM or IgD. And these molecules are acting as B cell receptors for the antigen. Is that right? No problem into this. Now, once the antigen comes, let's suppose star act on this. But others are not having any antigen. So this cell selected, very, very happy. And others are just sleepy. Is that right? Now this B cell has been selected because if antigen can fit into this B cell, it means within the nucleus of this B cell, there is already programmed to make anti-star antibody. Is that right? Now what will happen? On this particular B cell, which has been selected, right? Okay, let me make this B cell in a bigger way. This was IgM, which could react with star antigen. They will stimulate again, as there was TCR here. What is here? BCR. Here is CD4. There is no need of other molecule. Here was class 2. There is no processing here. Is that right? But there should be some signaling mechanism for the nucleus. There should be, isn't it? From the cell surface to the nucleus. There is special protein here. And these 2 proteins are called CD79. I think they were discovered too late. CD79. So we say surface immunoglobulins, once they are loaded with the appropriate antigen, these immunoglobulins move their tails and they activate CD79 and they give signal to the nucleus. Is that right? That congratulations. The antigen against which we were pre-programmed to make antibodies, that antigen is there. Now, look what happens. This activates a special type of gene, and when that gene is activated, that gene makes a protein, and those proteins are expressed on the surface of this selected B lymphocyte. So these special green molecules as green receptors are now expressed on the surface of this selected B cell. Do you think other B cells will have molecules like this? No. It means they are getting ready to be stimulated by something. It happens at times. Cells even get ready that someone should stimulate. And someone is really there. Who is there? Let me tell you. While this B cell is selected and it expresses its surface with special type of receptors, meanwhile, This has undergone enough clonal. Let's link the story number 1 and 2. This is occurring in the same lymph node. There were T cell stimulation and there is also B cell stimulation. Previously we talked about T cell was selected, then it clonally expanded. Once T cells have enough proliferated or multiplied, then suddenly they open another box of genes. They open a new box of genes. In those boxes, first of all, this gene is activated. And this gene produces a product. This gene produces a product. And this product binds here. What is happening now? T cells are helping in some way to the B cells. T cells are producing a product for which stimulated B cell has expressed the receptors. Is that right? And there is only one T cell, a lot of T cells. So all of them start producing product like that. Oh my God. So the super production of this unique green molecules, and these molecules are going to the B cells and they stimulate over here. Are you understanding? When these T cell produced molecules bind with the B cells, all these give message to the nucleus of which cell? B cell. You know what is the message? That similar antigen, this message to the nucleus, that similar antigen has been presented in a very proper way to T helper cells, and T helper cells are sending these special molecules. I will tell you what are these molecules. As soon as these green molecules go over there, there is a very special signal to one more type of, what is this? Gene. Okay, I will change the color of this gene. These all signals are going to this gene. And this gene in the which cell? B cell is activated. And once this gene is activated, there's a strong mitotic reaction into B cells. And B cell clones start expanding. And this particular B cell undergo extensive mitosis, and these cells number increase or decrease? Yes, the number increase. Do you know what is the name of this molecule? It is called B cell growth factor. B cell growth factor coming from the T cell, because it is leading to the growth of B cell. Or because it's also talk between the lymphocytes, isn't it? T lymphocytes talking to the B lymphocytes, this molecule can also be called interleukin. Actually, this is interleukin 4. Or technically we can say interleukin 4, which is produced by these cells, is acting as B cell growth factor. They are functionally B cell. So what is the function of interleukin 4? They act B cell as B cell growth factors and lead to the pre-selected growth of the pre-selected B cells. And there is clonal expansion of the B cells. Now in the lymph node, those B cells which have a capability to make anti-star antibodies, those B cells multiply. Right? Now, once these B cells have done enough multiplication, they start expressing another type of receptor. They start expressing another type of receptor. It means they are getting ready for another type of stimulation. Meanwhile, one more gene is activated into all these cells, T cells. And all these T cells start making another type of, yes, molecule. And that molecule will come and fit into these receptors, and these molecules are called B cell differentiation factor. What are these molecules called? B cell differentiation factors, or simply called interleukin 5. You see in the diagram interleukin 3 is missing. Actually interleukin 3 is also produced by the T cells, but that does not work in these. That works somewhere else. Later on you will learn interleukin 3 produced by the T cells work on the bone marrow, right? That is not relevant right now with us. right? Now you know interleukin 1 is from macrophage coming and stimulating the T cells. Now look, these T cells are helping the B cells. So this T cell should be called T helper cell. What should be this T cell called? T helper cell, right? Later on you will learn this very special type of T helper cell is called T helper 2 cells. I will tell you in later lectures where is the T helper 1. So this special type of T cell is called T helper 2 cell. So T helper 2 cell are producing interleukin 4 as well as interleukin 5. Interleukin 4 lead to proliferation of selected B cell. So interleukin 4 is B cell growth factor. Once B cells have enough proliferated, then interleukin 5 is being produced and interleukin 5 receptors are expressed by the clonally expanded B cells. And when B cells are stimulated by interleukin 5, all the B cells are stimulated by interleukin 5, they undergo differentiation. These B cells change into type of the cells. These B cells convert into oval cells with a nucleus with cartwheel shape, very prominent Golgi body, and all these B cells convert into cells like this. Are you understanding? Now what are these? These are the functional B cells. What is the function of these B cells? They will powerfully activate their machinery to produce such antibodies. You know, the antibodies which they displayed on the surface, right? And powerfully activate the genetic material to produce such antibodies, which are antibodies, anti-star antibodies. And they start synthesizing and throwing those antibodies out of the cell. So these become anti-star secretors, anti-star antibody secretor cells. Such B cells which are actively producing the antibodies, such B cells are called plasma cells. So what are plasma cells? Plasma cells are those B cells which are functionally very active and releasing antibodies. Resting B cell only express the antibody sample, but they don't secrete the antibodies. Are you understanding? But once they are properly clonally selected and clonally expanded and clonally differentiated, then such B cells which have been converted into plasma cells, they start releasing thousands of the antibodies every second. Thousands of the anti-star antibodies will be start releasing from the lymphoid tissue into body fluids. Is that right? No problem up to this? Now, these antibodies through the circulation eventually reach where was our bacteria. From the lymphoid system they will come into blood and antibodies reach over here, and these antibodies you will come to know, let's suppose this is bacteria, this is the antigen. Antibody will react with the antigen and then antibody will become active. As you put the bone in the mouth of a dog, it moves its tail. In the same way, antibody, as soon as it reacts with the antigen, its tail becomes active. And this activated tail can activate certain molecules called complement. And later on you will learn this activated complement will kill the bacteria. These activated complement will kill the bacteria. These complements are normally present in your blood. Liver produces complement molecules. Why we call them complement molecules? Because they complement the immune system. That's so simple. Right now, complement proteins are present in your blood. Right? When antibody fix on bacteria, right? Then antibody tail has an area which can activate the complement. And complements are activated at the surface of the bacteria. And complements, later on in lectures you will learn that activated complement produce the holes in the bacteria and bacteria dies. It's a very ruthless way to kill bacteria. Or complement can do one more thing. This is a bacteria, here was star antigen, and this is the, what will come here? Antibody. Antibody become active. Activated antibodies have receptors on the neutrophils and macrophage. So some neutrophil may come over here, and on the neutrophil surface, this is a receptor for, this is a neutrophil. Neutrophil have a receptor for the activated antibody. So antibody on one side is holding the bacteria, other side it is sticking to the neutrophil. Now, if neutrophil try to engulf the bacteria, can bacteria escape? No, it's a very bad way to kill someone. First you tie him so that it cannot escape, then you beat him. This is what neutrophil is, antibody is doing. On antibody, on one side hold the bacteria, on other side it holds the neutrophil or macrophage, and then bacteria has no way to escape. It will be caught by the antibodies. So in this way, caught by the phagocytic cell. In this way, antibodies facilitate the phagocytosis. When antibodies were not there, in the beginning of the lecture, antibodies were not there, neutrophils and macrophages were eating the bacteria slowly. But when antibodies are there, neutrophils and macrophage eat the antibodies eat the bacteria very rapidly. Just like that, this antibody is acting as a jam on the bacteria. And then neutrophils and macrophage love to eat it. It becomes more tasty. Is that clear? Such molecules which facilitate the phagocytosis of bacteria or other particulate matter, such molecules are called opsonins. What are they called? Opsonin. What is opsonin? A molecule which enhances the phagocytosis of particulate matter. Is that right? So what we see, that antibodies are produced, antibody react with the bacterial antigens, and either by activating the complement kill the bacteria or lead to very efficient phagocytosis of the bacteria by the neutrophils and macrophage. Is that right? Any problem up to this? Now, let us recap the lecture very rapidly, right? Okay, there's a diagram. You like to look to me or to diagram? You want I should explain or I should explain on the board? Last revision.
On the board.
You want on the board?
You explain.
I explain. Yes. Board. Okay. I think you are the final person. Yes. She just laughed it off. Yes. Yes. On the board. Okay. Let's suppose bacteria came. Now you have to tell me along with me. Bacteria came taken up by macrophage and by neutrophil. Neutrophil release free antigens, and macrophage take up the antigen and process antigen with class 2 molecule. Antigens processed with the class 2 molecule are presented to the appropriate T cell. Antigen react with TCR, class 2 react with CD4. Both of them activate CD3 and signal to the nucleus. Then the additional stimulations like interleukin 1 and its receptor B7 and CD28. And all these stimulations converge on the nucleus and activate the genes which are concerned with synthesis and release of interleukin 2 plus the genes which are concerned with the synthesis of interleukin 2 receptors. So selected T helper cells, now this is T helper 2 cell, right? It starts secreting interleukin 2 and interleukin 2 act on the same cell, and interleukin 2 receptors, once they are stimulated, they lead to stimulation of mitotic process in selected T cells. Selected T cells undergo clonal expansion, and eventually all these selected clonally expanded T helper cells will help the B cell to multiply and to differentiate into plasma cell. For this purpose, these selected and expanded clone of T cell will produce B cell growth factor, which is called interleukin 4, and B cell differentiation factors, which are called interleukin 5. Of course, if there was no clonal expansion, one cell cannot produce enough interleukin 4 and 5. That is why there was clonal expansion. Meanwhile, free antigen will sooner or later find one B cell which has such BCR which could recognize the or react with the free antigen. That particular B cell will be selected by the antigen, free antigen. That B cell will express the receptors for interleukin 4, and under the influence of interleukin 4 which is coming from the T helper cell, that special B cell will undergo clonal expansion. Clonally expanded B cell express the receptors for interleukin 5, and then these receptors are stimulated by interleukin 5 which is being produced by clonally expanded T helper cells. And under the influence of interleukin 5, these clonally expanded B cell convert into plasma cells. Plasma cells are very active cells, and they do a lot of antibody synthesis, and every second every plasma cell is releasing thousands of the molecules of antibodies. Antibodies come from the lymphoid tissue into general circulation, eventually reach to the site where the bacteria is there. Bacteria along with their antigens are coated by the antibodies, Antibodies activate the complement on the surface and complement mediated death of the bacteria occur. Or antibody bind with the bacteria, if no complement is around, antibodies tails are moving, they are activated, and activated tails are caught by neutrophil and macrophage, and of course bacteria is captured and it is efficiently phagocytosed. In this way, after the antibodies arrival on the bacteria, either bacteria are very ruthlessly killed by the complement activation, or they are very cleverly phagocytosed by the neutrophils and macrophages. That was an introduction to lymphoid system. Thank you.